All right, it looks like our attendance may be ticking down. We are just at half past the hour, so I think we can go ahead and get started. My name is Julianne Savat. I'm a genetic counselor and assistant professor with the GenomeConnect team, and I'm really excited for our webinar today where we're joined by Melissa Landrum. We'll be talking about how to search ClinVar and how to track a variant. But before we get into our topics for today, we wanted to just give a few reminders of our past webinars. So this webinar, like our past webinars, will be recorded, and you can access recordings from our prior webinars on the GenomeConnect YouTube playlist. We've listed them out there, so you can see those, and you'll be able to see today's webinar on that playlist shortly after today. We also have another webinar coming up in November at 3 p.m. Eastern Time, where Daniel Azaridi will be talking about how to track a gene in ClinGen. If you have already registered for that webinar as well, thanks for registering. If not, you can register to attend, and I'll get that link in the chat in a bit. For today's webinar, you won't be able to unmute yourself or come off onto camera, but we do want to spend some time at the end for some questions and answers. So you're welcome to use the Q&A box that should be at the bottom of your screen to tell us a little bit about what you want to hear more about or to ask specific questions. We do encourage you to use that Q&A chat for general questions about ClinVar, about genetics, genetic variants, and not for specific questions about your health or your genetic test results, just because this is going to be public. This is available as a recording. So before we get into sharing some information about ClinVar and really taking a step back and some background about variants first, we wanted to get a sense of who all is joining us today. So we have a quick Zoom poll that should show up on your screen just momentarily. And it will ask you to tell us a little bit more about why you're interested in learning about how to search ClinVar and how to track variants into ClinVar. In particular, is it for your own personal research, like if you found about variants on your own testing or a family member's testing? Is it for professional knowledge? So do you use ClinVar within your work? Are you a genetic counselor in the genetics profession and want to know a bit more? Is it both personal and professional reasons? Or is there some other reason why you're joining us today? I'll give it just a moment. All right, it looks like it's a split. So we have some folks here with us for their own personal research, some for professional knowledge, and some a mixed bag. So this is really helpful in how we might talk about some of these concepts, information, and what we'll share with you today. So thanks for sharing a little bit about who is on the Zoom with us. So to give you a sense of how we'll spend our time together, I'm going to get us started with some background information. So recognizing that it sounds like we have a diverse group of folks on the call in terms of their own personal experience or professional experience. We'll back up and talk a little bit about what genetic variants are, how they're classified, really broad strokes, just to make sure we all have a similar understanding, similar vocabulary. From there, I'll spend just a couple of minutes on GenomeConnect and our data sharing, really to help explain what is ClinVar, why do we share with ClinVar, what are the benefits that we see as a registry of sharing information there. And then I'll turn it over to our guest speaker, Melissa, to walk us through how to search ClinVar and how to track a variant in ClinVar. So first, getting us started with genetic variants and variant classification. So you all likely know this from your past experiences, but we're all made up of trillions of cells, and in each cell of our body is our genetic material or our DNA wound up really, really tightly. Our DNA provides instructions for our body, and each instruction is called a gene. Our genes provide instructions through the specific DNA sequence, which is the letters A, T, G, and C, repeated over and over again in different sequences. Our body reads those letters to make proteins. A genetic variant is a change in that DNA sequence, a change from what is typical. We all have genetic variants. Some impact our health, our development, put us at higher risk for health concerns in the future. Others are just part of what make us who we are. There are also different types of variants. So copy number variants are large missing or extra pieces of DNA, maybe hundreds of thousands of letters that are missing or extra. And then there are sequence variants, which are small, single, or maybe a couple letter differences in that genetic code. This will be important for when we get to how to search ClinVar, but we can name variants in a couple of different ways. So for sequence variants, or those small, single, or few letter differences, we can name them based on the DNA change, or we can name them based on how they change the protein. So remember, our DNA codes for proteins. So if we have a change in our DNA, it might change what that protein looks like too. If you see on a report that you have, or that a family member has, or are reading an article and see a genetic variant, if you see a C dot on your report, that's telling you what the change to the DNA is. So this C dot here. The C dot is followed oftentimes by a number. So that tells you exactly what letters changed in the DNA sequence. So in this example, we're talking about letter 1480 in the Cas gene. So if we start at letter 1 and count all the way to 1480, that's where we're looking. Then you often see a couple of letters. The first letter tells you what is the DNA letter that's typically there. In this case, most people have a G at that position, 1480. The little greater than symbol is our way of saying is changed to. And then the last letter tells us what letter that person has. So putting this all together, in this Cas gene, we're looking at the DNA because this is the C dot. We're at position 1480. Typically, people have a G. This individual has a T instead. If you're looking on your report or in an article, if you see a P dot, that means we're talking about protein. And this protein change typically has first an abbreviation like this GLN. That tells us what amino acid is typically at that position in the protein. The number tells us where we are in the protein. So at position 494, we often have this amino acid. And then if there's a change, we see that over here with this other abbreviation. So what this means is that this typical amino acid is changed to a termination or an early stop codon, making the protein shorter than typical. So putting it all together, P is protein, our reference amino acid, or what's typical, where's that position? So at 494, and what does it change to? So a couple of ways that we can name variants, and we have some additional videos and resources that go into this information in more depth if you're interested, but Melissa will come back to C dot and P dot in a little bit. So I wanted to tee up these things. So as I mentioned, some genetic variants impact our health, our development, risks for family members, while others do not. So anytime a genetic testing laboratory finds a genetic variant through testing, they work to classify that variant or try to determine if that variant causes health problems, developmental differences, increases risk. To classify a variant, they ask questions like, has anyone studied the effect of this variant in the laboratory? Have they looked in cells to see what's going on? If there are changes, if there are differences. They ask about if similar variants have been found. So maybe not this specific variant, but other ones that impact that same amino acid. Have we seen those in individuals with health concerns, developmental differences? Has the variant been seen in the general population, healthy individuals, people who don't have health concerns? Was the variant passed through the family? Is it new and unique in a child, or was it inherited from mom or dad, or do we see it in multiple generations? What's the impact on the protein? Is it changing just one amino acid? Is it making that protein shorter than typical? And has the variant been observed in anyone else with symptoms? They put all of this information together to come to a variant classification. They classify the variant into one of 5 buckets: benign, likely benign, uncertain significance, or sometimes you might hear this as a VUS, likely pathogenic and pathogenic. Benign, likely benign means that the variant isn't thought to impact how the gene works in the body, isn't thought to independently cause disease or increase risk. So enough information out there that we just pulled together to say, yeah, we don't think that this causes disease by itself. On the other end of the spectrum, likely pathogenic and pathogenic means that the variant's thought to impact how the gene works in the body, contribute to disease, to developmental differences, to risk. So enough information to say, yeah, we think this is impacting things. Maybe it provides a diagnosis or tells you about future risks. And then in the middle, this uncertain significance category is really for when there's not enough information about this gene or this specific genetic change, and it has a real uncertain or unclear impact on health based on the information. It may be there's just not a lot of information about that variant. It might be that we have some information that makes us lean towards benign, some information that makes us lean towards pathogenic, so really not enough information. Additional information over time can help inform uncertain results. So we might be able to, in the future, better understand that variant as we get more information. As we get more information, as that understanding changes, labs, clinics may reclassify variants. So we hope to reclassify those uncertain variants as benign or pathogenic as we understand more. And even our understanding of pathogenic and benign variants might change as new information becomes available. As I mentioned, this was a bit of a whirlwind of genetic variants, how we name them, variant classification, really because we want to get to how to search ClinVar, how to track a variant. So if you're interested in more information about these topics, we have a number of videos on our YouTube playlist that Caitlin so wonderfully already put in the chat. So you can take a look at those after today. So now shifting gears just a little bit. So what about ClinVar? What about data sharing? So we wanted to share a little bit about GenomeConnect's experience sharing data with ClinVar, how we think that that's valuable, and to provide a little bit of background about the ClinVar database. So as you may know, GenomeConnect is a patient registry where individuals who've had genetic testing can share their genetic and health information to make that information available through databases like ClinVar, which we'll talk a little bit about. Labs, clinics, researchers, they've been sharing genetic and health information with ClinVar and other databases for some time, but we felt strongly that patients could be a really valuable source of additional information. Patients are often experts about their own health history and can provide more detail about their health, their diagnoses, and access to that information can help us better understand what variants are tied to health and disease. So GenomeConnect's open to anyone who's had genetic testing. Individuals can send online and provide their health history via surveys. They upload copies of their genetic test reports. And then our team reviews their genetic test report and pulls out information about that genetic variant. So things like the gene name, that C dot, that P dot. So what's that variant name? We pull that information out in a structured way. So then we can, with our participants' consent, share that with the ClinVar database. Our guest speaker, Melissa, will provide more detail about ClinVar, but it's a publicly available database that holds information about variants and their relationship to health. It's public, freely available, and it's really used by healthcare providers, labs, and researchers who are trying to understand genetics and health. It's submitter-driven, so it relies on labs, clinics, even patient registries like GenomeConnect sharing data. So what a ClinVar record looks like, and Melissa will show this as well, but for any variant that's submitted, they first have a summary of the variant information. So some of the pieces that we've talked a little bit about, that gene name, that C dot, that P dot. So how's this variant named? And then if you scroll down from there, you can see submissions from labs, researchers, clinics, and even GenomeConnect if we've submitted on behalf of one of our participants. A couple of things to note about GenomeConnect's submissions. We don't classify the variant ourselves, but we're submitting the information as it appears on participants' reports. But we're providing a bit more detail than what's often included from labs, like information about that participant's health history, information about the variant's inheritance. In this case, it was new and unique to this individual. So just providing a bit more detail unique to our participant. As you can see, no identifying information is shared from our participants, but it's really focused on that health information, inheritance information. We focus on that information because it can be really useful in informing how we understand genes and variants. So it's unfortunately not uncommon for families to get an uncertain result through testing. And so the more information that we collect, the better we can answer questions about, is this gene causative of disease? Well, what about the specific variant? We can give that additional health information, inheritance information, all that can help us answer those questions. And then GenomeConnect participants also can have a couple of benefits back to them, like learning about additional research studies. We might learn about them because someone sees our ClinVar submission and reaches out to GenomeConnect, which we can then pass on to our participants. Participants can also connect with one another based on gene, and they can choose to receive genetic result updates from the GenomeConnect team. Those result updates also rely on ClinVar. So we identify them through our data sharing. So a participant might upload a report that says variant of uncertain significance from 2017. When we go to submit that to ClinVar, we might see that that same lab has submitted that variant with a different classification more recently, suggesting that our understanding has shifted, maybe more information is available. Now we classify that variant differently. We use our ClinVar submissions to note when this happens, and we give our participants the option to receive updates from us if we learn about changes like this. So in these cases, if the participant chose to receive updates from our team, we email them, alerting them that there might be updated information about their results and encouraging them to reach back to their genetic counselor, their doctor, to talk more about what that might mean for them. So ClinVar is important because for GenomeConnect, it helps us share information, get more information out there so we can better understand variants, their classifications. For our participants, it's also a way for us to identify updates that we can share back with them. So some participants, though, a question we often get is, well, I want to search ClinVar. I want to know how to use the database. I want to find my variant. And so that's why we have invited our guest speaker, Melissa Landrum, to share a little bit more with us. Melissa is the team lead for the ClinVar database at the National Institutes of Health. She earned her PhD in human genetics at the Johns Hopkins University, where she studied a viral vector for gene therapy. She came to the NIH 24 years ago, and prior to her work with ClinVar, she was a curator for another research project that provides reference sequences for genes, which are really valuable in clinical genetic testing and research. So Melissa, I think we could ask you lots of questions about the different hats you've worn, but today we'd love to hear more about how to search ClinVar and how to track variants. So I'll turn it to you.
Thank you, Jules, and thanks to the rest of the GenomeConnect team for having me here today, and thanks to everyone in the audience for joining us too. Next slide, please. So some of you may already be familiar with ClinVar. It's an NIH database of genetic variants and how they are classified for disease. So this refers to the types of classifications that Jules just described to you, where people look at the evidence for a variant and try to decide, does it not cause disease? It's benign. Does it cause disease? It's pathogenic, or do we not have enough information to know it's uncertain? Now, an important thing to know about ClinVar is that the NIH staff who run ClinVar are not the ones who are classifying the variants. So next click, please. Jules mentioned that there are lots of other groups out there who do classify variants as part of their everyday work. So this includes clinical genetics testing laboratories, research laboratories, curation groups who look at information, say, in the scientific literature or other public databases, and try and do that variant classification to decide, do these variants cause disease or not? There are also people like physicians and people who work at patient registries like GenomeConnect, who may not classify variants themselves, but they may be working with lab reports that have variant classifications, and they can add other information like the health history that Jules mentioned earlier. So many of these groups are dealing with genetic testing results that often turn up rare variants. And it's difficult to know how to interpret a rare variant. And so having an opportunity to share their data makes them much more likely to come up with a good interpretation of the variant. And so ClinVar provides a way for all of these groups to share their data. So these groups can voluntarily choose to submit data to ClinVar. My team takes the data, we do validation, standardizing the data. We group together data. So if multiple laboratories have seen one variant, we group all of their information together. And then next click, we package all of this information up and make it available for everyone to use as a free public database. Now, it is free, public, available for anyone to use, but that does lead me to a caveat. Next slide, please. Which is that I encourage you to look at ClinVar with your physician or your genetic counselor. And this is for two main reasons. One is that although ClinVar is available for anyone to use, it's primarily aimed at helping genetics professionals, so those working in clinical genetics testing laboratories, or say the medical geneticists and genetic counselors who receive those patient reports. So the language in ClinVar is often very scientific and uses some jargon from genetics, so it might not be immediately understandable to you. The second reason is that genetic data can be very complicated and there can be some nuances to it. So you may not get a cut and dried answer if you see something in ClinVar that you think relates to your variant. So I encourage you to talk with your clinician and get more information about what that ClinVar record might mean for you or your family member who's had genetic testing. Next slide, please. So with that caveat in mind, let's talk about how to search ClinVar. The homepage is a good place to start your search. I included the URL at the top of this slide, but you can also just Google for ClinVar and it should be the first result that you get, and it will take you to this homepage. So the homepage has a lot of links on it. If you're ever interested in digging in and finding out more about ClinVar, you can follow all of these links. But if you just want to do a search, you can focus on that search box that's at the top in the gray, and we can focus in a little bit more on that. Next slide, please. So this search box has a little bit of instructions in it. You can see it says search ClinVar by gene symbols, location, HGVS expressions, C. P. conditions, and more. So some of these things may be more or less familiar to you. These are all things that a scientist might use when they come to search ClinVar. But for this audience, I presume that many of you have had genetic testing or a family member has had testing and you're starting from a lab report. So yep, there you go. Thank you. So here we're looking at the lab report that Jules showed you earlier. And a typical lab report has a few pieces of information that will probably be very useful for you in searching ClinVar. Next click, please. So 3 of them are shown here, and next click. The first of these is the gene symbol. It's probably called out as the gene or the gene symbol on your lab report, but you'd be looking for something that's a short word or string of letters like this. It's usually all capital letters. It might have numbers as well. And then the next piece of information that would be useful would be the variant name. So this is the C. or the P. name that Jules just taught you about. So just as a reminder, the C. name tells you where in the gene there's a change and what that change is. And the P. name tells you where in the protein there's a change and what that change is. So any of these 3 things, the gene and either the variant names, are good things to search in ClinVar. And you can search for each of them on their own. That will work. So for example, you could search on just this C. name. And here we've done that search. And you can see in the bottom of the slide, I'm showing you the search results in ClinVar. The search results are a table. Each row in that table is a variant that's in ClinVar. And if you can read above that table, it says items 100, which means there are 100 variants in ClinVar with a change of C. 22, A to 7, A to G. So this search works. It gives you results, but you can see it's not very specific. And the reason is that C. description only tells you where in the gene to look, but you don't know which gene you're looking at. And if you look at that second column in the search results, that tells you what gene it's in. You can see it's in all different genes. So it's likely that there's 100 different genes that have this particular nucleotide change. So what I would encourage you to do on the next slide is search for both the gene and the CDOT name together. So you can just type them both into the search box. Make sure there's a space in between them so that we can tell that these are two different words that we should be looking for. And in this case, when you search for SCN8A and C.2287A to G, you get a single variant back. And in that case, it takes you directly to a variant page. So every variant in ClinVar has its own page with all of the information on it. Jules showed you that earlier. Here, we're just looking at the top of the page. Next slide, please. I'll also just quickly note that you could do the same kind of search with the PDOT name. So here we're looking at SCN8A and the PDOT name. If you're starting, from an article or something that only mentions the PDOT name, or if your lab report only has that, that's useful too. In this case, it also only gives you one variant back, so it's very specific. But if you ever have the choice between searching on the CDOT or the PDOT, you should use the CDOT because it's a more specific name for the variant. Okay, so let's look at the top of this variant page in more detail on the next slide. So as Jules pointed out, every variant page in ClinVar has that dark blue bar at the top that has a name for the variant on the left. And then it's followed by the gray boxes, and we call this the classification summary. So if you recall, ClinVar gets data from many different submitters who might be classifying variants. And so they may have different classifications of that variant, depending on what information they had when they classified the variant. So we want to give our users a summary of that information at the top of the page. So this is why we call it an overall classification. So overall, our submitters thought this variant was pathogenic or likely pathogenic. If you turn your head sideways, you can kind of see there's some labels in the boxes that say germline and somatic. So there are different contexts in which you can classify a variant. Germline variants are those that are inherited and they may be involved in genetic disease. Somatic variants are those that are not inherited. They may be more involved in cancer. But for the talk today, we're really just going to focus on germline variants that may be involved in genetic disease. Okay, so this is the overall classification that you see in ClinVar. If you were to scroll down this page, next slide, you would also find a table that looks like this. And this table shows you each classification that was submitted to us by a submitting laboratory. So in this table, each row comes from a different submitter. You can see the first 3 rows are from 3 different clinical testing laboratories. So you can see in the column called submitter, the names of these laboratories are Eurofins, GeneDx, and Invitae. And then the last row here is from GenomeConnect. The first column in the table shows you the classification from each of these groups. Jules pointed out earlier that the GenomeConnect submissions do not include a classification because they are not classifying variants themselves. They include information from the test report in the details of their submission. So let's focus on the first 3 rows that are from clinical testing laboratories. And you can see in that first column, they classified this variant as likely pathogenic or pathogenic. So this is individual classifications that we've gotten from different submitters. Next slide, please. So we take that information that the submitters have sent to us, and we use it to come up with that summary or that overall classification that you see at the top of the page. So we have code that follows a set of rules. And so, for example, it would look at the data that we have for this variant, and it would say, okay, our submitters thought this variant was likely pathogenic or pathogenic. Those are similar terms. They mean we think that this variant causes disease. And so the overall classification for this variant is pathogenic or likely pathogenic. So we calculate that value at the top of the page based on what our submitters have told us. And next slide, which means that if our submitted data changes, that overall classification can change. So let's say we get a new submission. Next click, please. And from here on out, this is a hypothetical example. We do not have new data for this variant. This is just for demonstration purposes. But let's say another laboratory classifies this variant, and with the information that they have, they classify the variant as uncertain significance. So again, we have code in ClinVar that would look at this and say, okay, now the variant has been classified as likely pathogenic, pathogenic, and uncertain significance. So uncertain significance is different from likely pathogenic or pathogenic. Uncertain significance means we don't have enough information to know if it causes disease or not. So in other words, these classifications don't agree with each other, they're in conflict. So when we would calculate an overall classification for this variant, we would call it conflicting classifications of pathogenicity. And this lets our users know that there's some uncertainty here. You need to dig into the data more to see what this means for disease or for a specific patient. So in this case, a new submission changed what the overall classification is. It might also change even if there's a single laboratory who has classified the variant. For example, let's say a laboratory first sees a variant and they decide it's of uncertain significance. Maybe a few years later, they see it in a second patient and they reclassify it. And now maybe they have enough information to say that it's likely pathogenic or likely benign. So a single laboratory may reclassify a variant, and that can change the overall classification. Or when we have multiple groups who've classified it, if they're in agreement or disagreement, that can change that overall classification. I'll also note that the direction and the classification can change in any direction. So in our example here, we went from some level of certainty that it causes disease to less certainty. So now people disagree. But it can go in the other direction as well. We can go from laboratories disagreeing on the classification to now they agree. Or like I said, a variant could go from uncertain significance to knowing whether or not it causes disease. But all of these changes are something that you might want to know about. If this is the variant that's been reported to you on your test result or a family member's test result, you probably want to know about this so you can talk about it with your doctor. So next slide. You can follow a variant in ClinVar so that you know when that overall classification changes. Next slide. Following a variant in ClinVar is pretty simple. So again, at the top of any variant page is that dark blue bar with the name on the left, and on the right are a set of links. And that second one, next click, it says follow. So if you just click on that link, and next slide, it will take you to a login screen. So you will log into NCBI. NCBI is the part of NIH that provides ClinVar. And we need you to create an account to use this feature. Creating an account is free. It's only used for your using certain features of databases at NCBI, so it's not used for marketing purposes or anything like that. There are several different ways that you can log in. Many of these are aimed at people who work within science, but some of them are more generic, like using Google or Microsoft accounts. So those might be good options for you to set up your account. So once you've created an account and logged into NCBI, next click, we will take you back to the variant page that you were just on, and you'll see that you're now following this variant. And if you do one more click, that link that said follow before now tells you you're following, so you know that you're now following this variant. So that's great. You're following with a variant. What happens next? Next slide. So going back to our hypothetical example here, this variant, let's say it goes from pathogenic, likely pathogenic, next click, to conflicting classifications. When this change happens, our code sees this change. We also know that you're following this variant, and next click, we will send you an email to let you know that there's been this change from pathogenic to conflicting classifications so that you can follow up with any actions that you want to take. So I will also note that this email, like all parts of ClinVar, it's written more in scientific words than in everyday language. So next slide, I'm going to use this as another reminder to encourage you to share the information that you see in ClinVar with your physician or your genetic counselor, whether that's information you see on a variant page, or if you get one of these emails that says your variant has now changed in classification, it's best to take it to your clinician to discuss with them to see what that might mean for you. Next click, please. If you ever Oh, sorry, there should be one more click in between there. If you ever have questions, thank you. If you ever have questions about searching ClinVar, following variants, anything about the database itself, you can email me and my team at the address that's shown there. But any clinical questions, it's important to take that to your doctor. My team is not a clinical team, but we can help you with questions about the database. And next slide. I finally just want to acknowledge the team of people at NCBI that works very hard to make ClinVar and all of this genetic data available. And with that, I will pass it back to Jules.
Thanks so much, Melissa. As Melissa said, if you have questions about your result, connect with your doctor or genetic counselor. There are some resources if you don't have a GC to find one in your area. That's where the QR code goes there. And we're happy to put that in the chat as well. So I think we have a little bit of time for some questions. As Melissa said, after today, the ClinVar team is happy to take questions about ClinVar and searching the database. We at GenomeConnect are happy to take questions about GenomeConnect, our data sharing participation. If you have them, feel free to reach out. We do have a couple of questions in the Q&A, Melissa, so I will do my best to tee us up for some of these. So one of the attendees asked what it means when you search your variant and find multiple disorders identified with the same variant. I know there are a few reasons for that and might be different reasons for different specific cases, but can you talk a little bit about that and why there might be multiple conditions listed in ClinVar?
Yes, that is an excellent question, and it comes up quite frequently for our scientific users as well. So genetics is complicated, and some genes may cause more than one disease, not typically in the same person. It may depend on where the variant is in the gene, or what specific part of the protein is affected by that variant. But there are many genes that, if they have a change, it can cause more than one disease. So sometimes you'll see for variants in the same gene, they are associated with different diseases. Sometimes even on the same variant, you might see that a laboratory has classified it for several different diseases. And most frequently, what that means is that the laboratory knows that gene is associated with several diseases, but they don't know enough about the patient's phenotype to know which disease that variant is really associated with. So there is actually a field in ClinVar that has a little explanation to explain that. But that's an excellent question. And as I said, it's of concern to our scientific audience as well.
Thanks, Melissa. We had a question that I think is related to GenomeConnect, so I'll try to take this one. So how does one get notified if the same variant is found in another individual? So as I mentioned for GenomeConnect, there's the opportunity to connect with other participants based on gene. This is historically something that within the platform, you can search for others based on that gene. If there are matches, send a message through the portal. This fall, we've recently changed our platform, so that feature is down for the moment, but we're working on getting that back up and running for our participants. If someone wants to connect with others based on gene or variant in the interim, they can reach out to us and we can see if there are any other individuals with that same variant, same gene. But in the future, we hope to have that up soon where you would be able to search on your own and take a look. So one of those features that undergoing some changes in hopes of making it easier to use and better to use. Another question for you, Melissa, is can someone follow more than one variant, or is it just one and done?
No, you can follow many variants. I'm not even sure we've put a limit on it. So absolutely, you can follow more than one. So for example, if you have more than one variant on your test results that you want to follow, you can absolutely do that. Or if we have any genetic counselors on the call, maybe you want to follow all of the variants for all of your patients, and you can do that as well.
Thanks, Melissa. We also have a question about, I think, sharing information. If we're sharing within GenomeConnect, sharing information, sharing symptoms, and will that let someone get an answer, clear classification? And this is a question we get a lot of, what information do we need to take that VUS to put it in a more definitive category of benign, likely pathogenic, likely benign? Unfortunately, there's no one piece of information that's going to solve all VUSs. Some are VUS because there's very little information out there. So we might need quite a few things to help us better classify it. Others are VUSs because we don't yet even understand the gene and the gene's role in the body. Some are VUSs because there's conflicting information. Some things that are pulling us towards pathogenic, some pulling us towards benign. So really, there's lots of reasons why a variant can be uncertain, and lots of opportunities for us to gain more information to try to better understand it. People also ask, well, if you give updates through GenomeConnect, when will I get an update? And there's no timeline for when we may see an update from our data sharing and be able to share that back with you. We do periodically look to see, are there updates? So it's not a one and done, but it's really as the landscape changes. And there's no, sometimes it's a month, sometimes it's 6 months, sometimes it's 3 years, and we're still in that uncertain category. So all depends on the specific variant. Melissa, a question for you. Genetics is changing so rapidly. New genes are being discovered, variants are being reclassified. How does ClinVar keep up with all of that? And it might be a bit because of how ClinVar is constructed, but maybe you can speak to that a bit.
Yeah, that is a great question. And you are right. Genetics moves fast. So part of the answer is that we don't actually have to keep up with all of the specifics. We're not the ones doing the classifications. Everybody in the clinical testing community in research, they're all doing this work and sharing their data through ClinVar. So the way we contribute to that is we've made the database fairly flexible. So in other words, we're not limited to people submitting genes that we already know cause disease. You can submit a variant in any gene to ClinVar, and you can submit any new disease to ClinVar, and that makes it very open to discovery for new gene-disease relationships. The part that we didn't cover today about somatic variants in cancer, that's another example where cancer testing is expanding quite a bit too. And so that was an area where we actually made some pretty significant changes to ClinVar so that we can keep up with that part of genetic testing.
Thanks, Melissa. It looks like we also have a user who has tried to search a variant, has specific, got a little lost in doing so, and was hoping for maybe a bit more guidance on the searching process. Okay, if I encourage them to reach out via that ClinVar email with more questions about that search?
Absolutely. You can contact us with any specific questions. We also have a nice YouTube video. You should be able to get to that from the ClinVar website, but it's a short, maybe 5-minute video about how to search ClinVar. That might also be useful, but absolutely, if you ever have a search that you think should work and you don't get what you expect, you can contact us and we're happy to help.
And I'll say we've helped GenomeConnect participants do the same. So ClinVar is a great place to go. We'll try to help you as well. One last question from a parent, I think, around there are many genes and variants that can cause the particular disorder that's in their family. Often amazed when they find others who have the exact same de novo variant, and just understanding kind of what that means and how often that happens. So I'm happy to take this one, Melissa, if that sounds okay. So yeah, I think we've had many participants, many families within GenomeConnect, within the rare disease community, who found others with similar variants, and sometimes similar variants, same variants, even though they're new and unique to their children. So it does happen, and it just means that those two children have the same genetic change that happened very early on in development or in sperm or in egg. And those are ones that are impacting how that gene is working in the body. So I think families love when they can connect with others with the same genetic cause, so same gene, but certainly families who've been able to connect with same variant too. Of course, even individuals with the same exact variant might not have all of the same symptoms or have all of the same health differences, developmental differences. And it's because that one single genetic change is just one of many other genes, many other pieces of genetic material that make up those children, and lots of other factors that go into health development, all of those things. So it does happen. And sometimes it could be just by chance. Sometimes we know variants are recurrent, meaning that we sometimes see them multiple times just because that's a maybe part of our genetic material that's tough for our cellular machinery to copy, for instance. So it can happen. All right, looking at the time, looking at our questions, I think we've answered everything. So I just want to take one more moment to thank Melissa for sharing expertise on ClinVar, how to search based on that CDOT, PDOT, and gene. Welcome other questions via email, and have a lovely rest of your Wednesday all. Thanks for joining us.
Thanks, everyone.